Hello, good afternoon. I hope you have studied very well up to the question number 110 in the previous question in the previous video. So let us start with the next question. Apart from the 110 question, 111 we have to continue from here. So here the question number 111 is saying given below are the stages of the life cycle of the pteridophyta. Okay, please we have to arrange the following stage in the correct sequence. Okay, so as you have studied about the correct sequences, so we can say the first one should be the meiosis, because if the meiosis, the division will start, because it is going to show the mitosis and the meiosis division. So first meiosis division will be first mitosis and then meiosis. Mitosis not given here, so we'll go with the first statement. That means the B should be the on the first stage. Okay, and then after we can say the prothallus stage will come. Okay, this should be on the second. So B and then A. B and then A given in the two options. Rest two will become eliminate. And then D or E either. So formation of the archegonia and the antheridia in the gametophyte. Okay, this will be on the third stage, and then only the anthrozoid will become transfer. Okay, so first of all the formation and then the transfer. So first D and then E. So D and E given in the first option. The next question number 112 is saying cardiac activity of the heart is regulated by the nodal tissues. So nodal tissues, in the sense they are trying to say about the conduction system. Okay, in the conduction system we have seen that the conduction system is having the four stages. It is started from the AC node, which is present on the upper right corner of the heart, and then it will give the signal to the AV node, then bundle of His, and then finally it will give the signal to the Purkinje fiber. This Purkinje fiber will give the signal to the ventricle muscles. Okay, then the contraction will be there. Ventricle systole will occur. Start from here. Okay, so here the cardiac activity regulated by the nodal tissues or the special neural center in the medulla oblongata. It is present into the medulla oblongata, but it will control by the pons area, because in the pons we have seen the apneustic and the pneumotaxic center. Okay, two centers are present into the pons area, apneustic and the pneumotaxic center. Apneustic and pneumotaxic center. Okay, so these two are present into the pons area, and rest centers like respiratory rhythm center, cardiac activity center, vomiting center, sneezing center, that all present into the medulla oblongata. Okay, but they all are controlled by the pons area, these two terms. Okay, so here let's see. So cardiac activity will be controlled by the conduction system as given by the nodal tissues, the neural center which is present into the medulla oblongata, and then after adrenal medullary hormone. Adrenal medullary hormone was epinephrine or norepinephrine, also known as catecholamine. Catecholamines. Okay. So this one, and cortical hormone will not be regulated the cardiac activity. So A, B and C given in the first option. The next is the which of the following organism cannot fix the nitrogen. It is again memory based question. So we can say only the Volvox is there, which is showing the no fixation of the nitrogen. Rest others are able to do the nitrogen fixation. So they all can do, but the Volvox cannot do that. So the option number second will be the answer. Then the 114, the transfer of the RNA and the ribosomal RNA do not interact with the mRNA. Okay, so we can say transfer RNA and ribosomal RNA, if they are not interacting, then how the protein synthesis will be there. So for the protein synthesis, tRNA and the mRNA both are very important, and they are saying it is not important. So it is incorrect. RNA interference takes place in all the eukaryotic organisms as a method of the cellular defense. It is a natural method. So first one is incorrect, but second will be correct. Then next is they have given the PBR 322 here, and they are saying here is a plasmid, not only the PBR. Here will be the plasmid, and this plasmid having the tetracycline resistance gene, ampicillin resistance gene, and EcoR first. One more we have seen CLA Hind. Okay, CLA first and Hind third. And in the EcoR first, they are showing that beta-galactosidase gene is inserted. Because of the laxate gene, the beta-galactosidase gene inserted. So because of that, the EcoR first site will become inactive. Because of the insertion of the beta-galactosidase gene into the EcoR first site, so the insertional inactivation will be there for the EcoR first. Okay, it will show the insertional inactivation. Okay, so because of that we can say here what type of the color colonies will be there in the if the beta galactosidase was present, so it was interact with the X galactose which is present already onto the plate. Okay, but if it is not, if it is insertion inactivation will be there, so because of that the beta galactosidase will not show the synthesization, and X galactose will become alone and will not interact with the beta galactosidase, so the colorless colonies will be there. So we have, I show you on this plate in the side, plasmid was having the LAC-Z gene. This is the LAC-Z gene. So LAC-Z gene is responsible for the beta galactosidase synthesization. Beta galactosidase. Okay, and this beta galactosidase, by transferring into the E. coli, this plasmid will transfer into this E. coli. Okay, okay, just 1 minute. Here will be the E. coli cell or any other cell, transferable cell, and then we are transporting this inserted gene into the beta galactosidase will be synthesizing here, and this we will transfer it onto a plate, Petri plate, and on this Petri plate we kept the media, and on this media we are keeping the X galactose. Okay, it is X galactose chromogenic substances. Okay, so if it is present and this beta galactosidase is also synthesizing from here, so because of this the beta galactosidase can interact with this X galactose, and they both together will give the blue color colonies. Okay, blue color colonies was there. But if suppose if we are inserting our gene of interest into the LAC-Z site gene in this area, so because of that beta LAC-Z gene will not express, and beta galactosidase also will not express. If it will not express, so it will not interact with the X galactose, so the color of the colonies will not become blue, it will become colorless. Okay, it will become colorless. Colorless in the sense it will, we will call it as a white. White is not having any color, so we will say it will become white colonies. So this is the experiment by which we can do the selection of the with the help of the colonies. Okay, so that is what they have given in the question. So here they are showing the gene of interest is inserted into the by using the eco-ARPA in the beta galactosidase. So because of that, what they are saying in the question, in the above representation, plasmid, an alien piece of the DNA is inserted at the eco-ARPA site. Okay, so we have inserted the gene of interest in this area, eco-ARPA site. Which of the following is strategy to be chosen to select the recombinant colonies? If the colonies will become recombinant, so because of the recombination, what will happen? Beta galactosidase will not synthesize and it will not interact with the X galactose. Okay, so no interaction will be there. So because of no interaction, there will be no blue color colonies. Okay, so they are saying using ampicillin and tetracycline containing media plate, no. Blue color colonies will not produce. White color colonies, yes, it will select. Blue color colonies grow on the ampicillin. We are not using antibiotic. Okay, so we can say the third one could be the answer, because they are saying colorless colonies will be there because of insertional inactivation. Okay, I hope this experiment would be clear to you. Now let's see the question number 116 is saying which of the following genetically engineered organism was used by Eli Lilly to prepare the human insulin. For human insulin, what they have used, they use the bacteria. Okay, and then the transferable or non-transferable, what type of the class is there? Classification of the enzyme you have studied into the biomolecule. 6 classes. Okay, 6 classification of enzymes we have studied, and in the sequence you have to learn it. Okay, that is very important. So what type of this reaction, in this reaction is given in which type, which class of the enzyme, that you should know. That is very important. This is given into the transferase type. Okay, it has been given into the transferase enzyme. Okay, so G, they are showing a group other than the hydrogen, as is the substrate, as dash is the another substrate, so it is all happening into the transferase. Okay, so here, and then next 118 is saying find the statement that is not correct with regard to the structure of the monocot step. So here it will be the memory based question, because hypodermis is parenchymus, which is not the monocot. Then correct sequence of the event in the life cycle of the bryophyte. In the life cycle of the bryophyte, if you have learned, so we can say from the 5 points, which could be the first stage in the life cycle. Okay, we can say the attachment is important. First of all, the attachment should be there for the gametocyte to the substratum, and then after okay, so and then after what could be the B is there. Okay, so the first and fourth will become eliminate. Because of that, we can choose either B, E, A, B, E, A similar. Okay, so the B and E and then A. Okay, after fusion of the anthrozoid with the egg, what will happen? Whether the formation of the sporozoite or the reductional division. First of all, if the gametocyte is present, anthrozoid with the egg fusion will be there, means the fertilization has been takes place. So because of the fertilization, what will happen? The new spores will become formed, first of all, and if that will happen, then then we can say okay, so the reduction division to produce a haploid spore. Okay, so haploid spore because of the sporophyte. So first the sporophytes will form, and then the formation of the spores will take place. Okay, so this will be the fourth one, this will be the five one. C first and then D. Okay, so here D first and then C. So third statement, third option. Okay, the next is what are the correct about what about the chemotherapeutic agents, drugs and interferons, chemotherapeutic drugs and the leukemia, means they all are from the human health and disease chapter. So it is related from the chapter. So we have to correctly check that what they are saying about these drugs and interferons, and then we have to find out the correct answer for that. Okay, so the first one is saying that computed tomography, CT scan and the MRI, magnetic resonance imaging, detect the cancer of the internal organ. Is it so? Internal organ we are using for the detection. Okay, and then chemotherapeutic drugs are used to kill the non-cancerous cells to the cancerous cells, not the non-cancerous cells. So B should not be there, A should be there. B should not be there, A should be the answer is here. But even we should know about the other statement also. The alpha interferon activate the cancer patient immune system and help in destroying the tumor. Alpha interferons is just like we can see the example, like if someone home is having the fire, like something is happened, because of that the fire will become burst in the home. So because of that fire or the burning in the house, some smoke will be there in the nearby area outside the house from the windows and the doors. So because of that smoke, the nearby homes will become activated, they will close their door. So that smoke is actually the signal for the nearby houses. In the same way, the cancerous cell will produce some alpha interferons, some signaling molecule, and these signaling molecules will become the signal for the nearby healthy cells. So they will make the membrane hard. So the cancerous cells or the damaged cell will not able to give the damaged part or the damaged protein to the nearby cells. That damaged protein will not able to enter into the nearby cells. Okay, so because of that we can say alpha interferon is just like a signaling. So it would be correct. So chemotherapeutic drugs are biological responses modifier. Okay, and in case of the leukemia, blood cells count are decreased. That is also, but the combination would be not given for all, they are asking for it. So in the fourth option, A and C both are absolutely correct. Then 121 is saying centromere, cilium, cisternae, cristae or the cell membrane. We knows very well cristae into the mitochondria, cilium or the cell movement with the help of the cilium. Okay, cell division, and cell membrane will be phospholipid layer, and centromere will become the cell division. So A should be 2, B should be 3, A2, B3. Okay, and it given in this one. Then chlorophyll A, B, xanthophyll and the carotenoid. So here chlorophyll A will become the yellow green algae. Okay, blue green algae. Okay, so blue green algae A3, where they have given the A3. Okay, so A3 given in these two will become eliminate, and then after B we should choose. So B is saying chlorophyll B. Okay, chlorophyll B will be into the yellow green. So yellow green, B should be 1. So B should be 1 here given in this option. Okay, the next is correct sequence we have to find out. In human pregnancy, the major organ of system are formed at the end of the 12th weeks. So at the end of the 12th week we have seen the formation of the major organ. Okay, so that is what it is correct only. In the human pregnancy, major organ, this is the complete paragraph given in the human reproduction chapter for the in the pregnancy stages, 9-month pregnancy. So which organ will form in which stage, in which month or the trimester or the second trimester. So you have to learn all these stuff. I will share into the description box about that complete slide, so it will be easy for you to learn it. Okay, so let's see about this one. A is correct only. Human pregnancy major organ system are formed by the end of the 8th weeks. No, here they are saying on the 12th weeks and 3rd month. Okay, so here 8 weeks incorrect. B should not be there and A should be there. Okay, answer is here. But let's see others. Heart is formed after the 1 month of the gestation. Okay, human pregnancy limbs and the digit develop by the end of the 2nd month. Okay, and human pregnancy appearance here observed in the 5th month. Correct. So the option will be the 4th one. Okay, now the next is question number 124. In the seeds of the seers, the outer covering of the endosperm separate the embryo by a protein-rich layer called as what? So it is a memory-based question again. So we know alluron layer is the layer which will be protein-rich layer. Then next is from the excretive product and the elimination. We have to find out the correct one. Water cannot be reabsorbed here for the secretion. Water should be reabsorbed by the blood vessel. It should be here. It should be here. It should be here. Okay, as given here, but not in the PCT for the secretion. Okay, so could not be there. And NH3, okay, ammonia should be there. Potassium they are saying here. Okay, so then ammonia and this proton ion and potassium and the proton ion, okay, it is excreting, and all the blood vessels will be reabsorbing from here, from the DCT and the PCT. These are the good molecules. Okay, we can say this complete diagram of the NCERT. Here the ammonia is secreting, okay, potassium is, proton is secreting, but NaCl should not be secrete. It should be reabsorbed. It should be above like here. So it could be incorrect, and here the water again, so incorrect. So the correct answer should be 2nd one. The next, what is here in the bioreactor which is used for the foam breaker. C is used for the foam breaker because it is a blade. Okay, which is blade which is help in the mixing as well as foam breaking. Mixing and breaking the foams. Breaking foams. Okay, so it should be the 4th one. The next is assertion reason based question. So here a typical unfertilized angiospermic embryo sac at maturity is 8 nucleated and 7 celled. Okay, the egg apparatus is a 2 polar nuclei. It is not polar nuclei. It will be incorrect. Okay, so we can say 127 will become the 3rd option. A true but reason false. Then the next is a specialized membrane structure in the prokaryotic cell which help in cell wall formation, DNA replication and the respiration. So we can say in this one, like mesosomes is actually applicable for all these stuff. Cell wall formation will be there because of it, and DNA replication and respiration also will be there because of it. It is a mitochondria. It is because of in the mitochondria. Chromatophore will not be there. Endoplasmic reticulum will be the part of the outer membrane of the nucleus. So it is also not. So correct should be the 1st one. The next is which of the following are the post-transcriptional event in all eukaryotic cell. Post-transcriptional means after the transcription what has happened among these all. So we have to read it properly and then we have to find out the answer. Okay, so the transport of pre-mRNA to the cytoplasm prior to the splicing. After splicing it can go to the cytoplasm. Before splicing cannot go. So A should not be there. Okay, so A should not be there. And removal of the introns and the joining of the exons. That is correct. That is actually the splicing process. So that will happen after the transcription. But before the transcription, it cannot leave the it cannot leave the nucleus or the endoplasmic reticulum. Addition of the methyl group at the 5' end of the hnRNA. Okay, and this is the, we can say, addition of the methyl group will be helping to the hnRNA for the synthesization of it, of the transcription. Addition of the adenine residue. This is known as a polyadenylation. Okay, so this could be there. And base pairing of the 2 complementary RNA. No, double stranded RNA will not be there. 2 separate RNA will be there, and then the protein synthesis from the 2 different strands. So E should not be there. So BCD would be the correct answer. BC but not E. Okay, so we'll go with the 2nd one. And what is the pattern of the inheritance of the polygenic traits? Polygenic means more than one gene will become expressing here. So that was all because of the non-Mendelian inheritance pattern in the genetics we have seen. Then which of the following enzyme contain the heme as the prosthetic group? In the heme, what is the prosthetic group? Catalase. Succinic dehydrogenase will be the endoplasmic reticulum having the electron transport chain, the second part of the electron transport chain. That was the because of the succinic dehydrogenase, and it is also the example of the competitive inhibitor. Competitive inhibitor. In the competitive inhibitor, succinic dehydrogenase was inhibited by the succinate, by the some inhibitor. This dehydrogenase will become inhibited. Carbonic anhydrase, we knows very well, it will convert the CO2 and the water into the bicarbonate ion, H2CO3. That is a complete reaction which is occurring into the RBC. And Rubisco will help into the C3 cycle. So it is also will not be there. But in the among the four, catalase is only responsible for the cofactor, which is a catalase, which is the here prosthetic group. And then 132 is saying each of the following characteristics represent a kingdom proposed by the Whittaker. Arrange the following in the increasing order of complexity of the body organization. Okay. So for the body organization, the complete sequence we have to find out. Let's see about it. The first one should be multicellular heterotropic with the cell wall in the chitin. This is about the fungi. Okay, heterotropic tissue, organ and organ system and body organization. This will be the animalia. Okay, it will become the animalia or the animal kingdom. Prokaryotic means monocytes. Okay, means single cellular organism. And eukaryotic, we know eukaryotic with the cellular type of the organization. That is also there. But here among these four, from the starting to the last one, means Whittaker have given the kingdom system. No? So for Monera, Protista, fungi, animalia. So like that we have to make a sequence. So for that, the first one should be the Monera. This is the Monera. Okay, C would be the on the first one, Monera. And then after Protista, eukaryote, this will become the Protista. Okay, so this should be on the second one, C and E. These two will become eliminate, and then A also is already there, fungi, Monera, Protista, fungi. So first Monera, Protista, fungi, then animalia, MPFA. Okay, so then after A should be there, and after that among all this eukaryotic autotrophic organism, tissue, organ, level organization that we have seen. Okay, and then finally in the last, animals should be there. Okay, animalia, complex organism, that is animalia in the last. So B should be there in the last. So second one. Then who is known as the father of the ecology in India? So father of the ecology, Ramdev Mishra. Okay, then Alfred Hershey and Chase experiment, euchromatin is actually loose form, heterochromatin is the condensed form. So loosely packed and lightly stained, euchromatin, heterochromatin, densely packed. Okay, then Frederick Griffith, Streptococcus pneumonia, Hershey Chase DNA is a genetic material. So A should be the fourth one. Okay, and B should be the 3, A4, B3. Okay, given in this third statement, and matching I have given you already. Neoplastic. Acha, one more question remaining down, I think. 134. Okay, 135. Okay, 135 is neoplastic characteristic of the cell refers to what? Neoplastic characteristic will be the mass proliferation, rapid growth of the cells, invasive and damage to the surrounding tissues or those confined to the original location. So among these we have to find out the correct sequence. Okay, so here the first one, obviously the proliferating cells will be there because it is a neoplastic cells characteristic. So new cells will be forming. So because of the proliferation only it can happen. Okay, so the A should be there, and B is also the rapid growth. Okay, cell division and growth will be there, and then invasion and damage to the surrounding tissues. Okay, that is correct. The confine of the original location, it will not confine to the original location. No confinement on the original location. So D should not be the remaining 3 will be correct. So A, B, C. Then the DNA fragment extracted from the gel electrophoresis can be used in the construction of recombinant DNA. Okay, we have seen, just 1 minute. Okay, so DNA fragment extracted from the gel electrophoresis can be used in the construction of the recombinant DNA. Okay, so we have seen in the gel electrophoresis, gel plate, agarose gel plate we have seen, in which the DNA fragment will be there, and the complete fragment we cannot take it complete fragment, so we have to find out, take the complete piece of the gel from the gel. okay, like this. And if we will take the gel, complete gel piece, so from that we can take the complete fragment. We cannot take the fragment in the hand. So the fragment can be extracted from the electrophoresis by using the construction of the recombinant DNA. Okay, so this is correct. Second is a smaller size fragment are observed near the anode. This is agarose gel plate. Here we are keeping the negative and here we are keeping the positive. Yani ki anode is on the downside and cathode should be on the upper side. And DNA fragment we are keeping it here. Okay, and we know DNA will uploading and DNA is a negative charged. DNA is negatively charged. So DNA is negative charge, then how the fragment will move to downside? Because the negative charge will interact with the positive charge and it will move towards the downside. So because of that it is moving downside, so the smaller fragment will become very faster. This will become the smallest and this upper one will become the largest, okay, or heaviest. Largest and heaviest would be on the upper side, smallest and lightest would be on the lower side. So the smaller side DNA fragment observed near the anode, okay, while the largest will become towards the well, yani ki upper side. So it will become in the agarose gel. Okay, that is correct. So both statements are correct one. Then adenosine, adenolic acid, adenine and alanine. Alanine is what? Alanine is a amino acid. A should be fourth one. Okay, what they are asking? Okay, correct matching. Okay, so A fourth not given in anyone. Okay, let's see others. Adenine is what? Adenine, adenolic acid nucleotide. Okay, so B should be here too. Okay, B ka 2 we can say, B2 in these two. So these two eliminated and A3 given here. A already nucleoside, adenosine nucleoside. Okay, so sorry, A should be 2. Sorry, A should be 3. Okay, B2, A3 and D should be alanine is what? Alanine they have given as a amino acid or the nitrogenous basis. D should be fourth one. I was seeing A should be fourth one, but okay, sorry for that. D should be fourth one. So here D fourth. So we can say it could be the third. Then reductive division for the human female gametophyte starts earlier than that of the male gametophyte. In the female gametophyte will be starting earlier in comparison to the male. That is what they are saying. Okay, so it is occurring fastly and rapidly and sooner. And gap between the first meiotic division and the second meiotic division is much shorter for the male compared to the female. In comparison to the female, it will be happening. And because of that reason is also that it could be the association reason based question. And the first polar body is associated with the formation of the primary oocyte. Primary oocyte will give the division, two unequal division. One will become the primary oocyte, primary polar body, and another will become the complete secondary oocyte. This was the secondary oocyte. Okay, so this was the primary oocyte, and primary oocyte was showing the meiosis 1 division. Okay, because of that the one polar body and one complete secondary oocyte was formed. So C should not be the first polar body associated with the secondary oocyte, not the primary oocyte. So C should not be there. A and B together given here, and LH lead to the disintegration of endometrium and onset of the menstrual bleeding. Not because of the LH, menstrual bleeding because of the low LH and both FSH, LH and FSH both, not because of that. Then living members of the cyclostomes are, living members of cyclostome will be always on the upper surface, not the inside. So endo not there, it should be ecto. Okay, then assertion is saying the primary function of the Golgi apparatus is to pack the material made by the endoplasmic reticulum and deliver it to the intercellular target and outside the cell. So the main function of the Golgi apparatus is to pack the material in the, and this is the nucleus, endoplasmic reticulum, and here will be the Golgi apparatus. In the Golgi apparatus we are having the two phases. This part will be the trans phase and this part will be the cis phase. So the ions or whatever protein will synthesize, it will come and by entering into the Golgi body from the cis phase, it will enter into the another part, and then by crossing from the trans phase it will leave the cell. If it has to go to another cell, then it will leave this cell and it will go to the another part of the cell or another cell for its functioning. Okay, so that is what they have given here, outside by the trans phase. And the vesicle content, this is what happening by packaging itself into the vesicle. Okay, this vesicle will go outside. Okay, like this. So vesicle containing material made by the endoplasmic reticulum fuses with the cis phase of the Golgi body, and the modified the release from the trans phase of the apparatus. As I told you already, the same thing they have given in the reason. So that is why it would be correct, as well as the explanation also will be the correct.